I'm Jessica Kramer. I'm part of the Chemical Analysis and Life Detection group at JPL. Our group is interested in building instruments and methodologies to search for chemical signs of life on other planets. One particularly interesting target, and the reason we're all here in this room, is Enceladus. The chairs of this session did a great job in motivating all the different lines of evidence that Cassini found that showed that there could be an active ocean-rock interface in Enceladus's ocean. This is a really exciting discovery and opens up a lot of questions as to the habitability of Enceladus's ocean and the chemical composition, and really is a nice motivator for sending another follow-on mission to pick up where Cassini left off and really get more specific chemical analysis done on the compounds in the pollutant water. One particular target that we're interested in going to look for are amino acids. Amino acids can tell you a lot about what's happening in the planet because they can be generated abiotically through geochemical processes. They can also be an indication of prebiotic chemistry, and then also maybe point to the presence of extant life. But it's not exactly enough to just go and find amino acids because they can be made abiotically, and in fact there is a lot of overlap between the amino acids that are formed biotically and those are formed abiotically and those that are used in biotic systems, not surprisingly. So instead, it would be good to go and bring a technique that can survey all the different amino acids in the sample. And once you have that, and you have that information, you can start to look for patterns that can indicate the presence of abiotic or biotic chemistry. One of those patterns is what type of amino acids are there. In particular, histidine is only made through biotic chemistry, so if you were to find that somewhere, and you know we didn't bring it with us, then it would be a good indication that something other than abiotic chemistry was taking place. The next pattern you can look at is the relative abundance of the amino acids you find to glycine. Glycine is the smallest and easiest to synthesize amino acid, so abiotic chemistry makes a lot of it with respect to the larger, more complex amino acids. But life needs complexity, so relative to glycine, life uses more of those complex amino acids, so you see the higher abundance of those in biotic samples. And then finally, chirality. In abiotic systems, the left and right-handed versions of the amino acids are generally created in a 50-50 mixture. So if you were to see any sort of an antimeronic excess or homochirality, that would be a really good indication that it's not just abiotic chemistry. So in order to go and search for those signs, you need a technique that can take a sample that has a mixture of all those amino acids, separate them out so that you can individually identify and quantify them. And the technique that we think is the best is capillary electrophoresis. It's a voltage-driven separation, and it's all liquid-based, so which is really good when you're going to go study water or ice. It also has very low limits of detection, down to nanomolar or parts per trillion, when coupled with laser-induced fluorescence detection. And it's miniaturizable. So this is the big system. The commercial system, it's about the size of a dishwasher, and this is one of our portable systems, the chemical laptop, that is about the size of a shoebox. So if you're interested in learning about how we're shrinking that technology and making it more portable, go to Fernanda's talk tomorrow. So CE works by filling a small bore capillary, about 50 micron inner diameter, with a conductive solution. You can then inject your liquid sample directly onto the capillary, and when you apply a voltage, everything in that plug starts to separate based on its size to charge ratio. So you start to get a separation of all the different compounds in solution. Is it playing? As those separate, they move down the capillary and they move past the detector, in our case, a laser. And so you can collect a signal versus time plot we call an electropherogram that can be used for identification and quantification of the different species in solution. So this is our electropherogram from a method that we made. This method targets the 13 most abundant amino acids found in both abiotic and biotic samples. There's a lot of overlap there, and so with these 13 amino acids, you can look for those biosignatures I talked about previously. So you can see we have 13 amino acids, but we also are able to, man, I'm bad at that, separate the left and right-handed versions of the amino acids for 5 different amino acids. So we can look for that chiral signature as well. So So back when I published the method in 2017, our limits of detection were between 5 and 100 nanomolar. It's pretty good, but we've been able to get them down even lower to between 1 and 25 nanomolar, depending on the amino acid. And this is really great because it puts us in the range of what we see for Earth analog, like subglacial lake environments for free amino acids. So we're in a good place to be able to detect free amino acids in ocean water. Another great thing about this technique is it makes it really easy for end-to-end analysis of liquid samples. So if you have a liquid sample, all you need to do is mix it with some reagents to fix the pH, add your fluorescent tag, and then analyze that directly. Because we get those low limits of detection, and because we're doing separation, you don't need to do any additional pre-concentration or desalting. In fact, we're able to look at a variety of different samples. Here I see monolake water and Atacama soil extract, and we can see very low concentrations of amino acids in these samples, even though there's a variety of different salts and other things in the matrix. So because of the capabilities of this method, it was selected as part of the analysis suite on a New Frontiers proposal called Elsa. This is the Enceladus Life Signatures and Habitability Mission. And it's a multi-center collaboration. It's a flyby mission, so it would fly through the plumes, collect sample. That sample would be transferred to a sample handling system. And I think Splice from Ames is going to talk about that next in this session. That sample handling system also includes the reagent storage needed to be able to run the capillary electrophoresis test and analyze for those chiral amino acids. And Elsa didn't get selected this time, but I think there will be a second proposal because I think it's a really important mission. And so in order to get ready for the next proposal, we've been demonstrating on the stability of the hardware and the chemicals under spaceflight conditions in order to increase the TRL, or technology readiness level, of this technique. I've been focusing more on the chemical side of things, but if you're interested in hardware, go see Nathan O'Borney's poster tonight. He's going to talk about making radiation-tolerant hardware specifically for the detection side of things. The chemical stability is really important too, and it's something that people always have questions about because we're bringing these like complex, mostly organic molecules along with us, and they're worried about them falling apart. So the important chemicals for this test are the fluorescent dye. This is how we label our amino acids and create a fluorescent compound, then that's what the separation is designed to detect. And then in order to get that really nice chiral resolution, the resolution of all those amino acids at once, we do need a kind of complicated background electrolyte. That's the conductive solution that fills the capillary. It's a mixture of 3 powdered reagents, and it's mixed with an aqueous solution of 6% acetonitrile. So all that needs to fly in order to be able to perform this assay. So radiation is the big problem. Traditionally, organic chemistry isn't great under radiation, so we were pretty worried about this, and to choose the worst case scenario, we chose Europa. This is far worse than anything we'd see in Enceladus, but still, if we can do it under these conditions, then we can do it anywhere. So we chose the number from the Europa Lander Science Definition Team report of 300 kilorad. The first thing we tested was our dye, so we exposed the dye to 300 kilorad, and we were actually kind of shocked that it was fine. Not only did it retain its fluorophore, so it allowed us to be able to detect it at the same concentrations as non-irradiated dye, but it retained, I'm not going to mess this up, it retained this leaving group. So this is a pretty labile chemical because it's needed to react with the amino acids, but it was able to retain the labeling efficiency. And then even better than that, there were no additional peaks formed. So this is a fluorescent molecule. So when you start to irradiate it and it starts to degrade, it can turn into other fluorescent molecules that can interfere with our separation. And we didn't see that, so this was really great. We also irradiated the background electrolyte, and we found that as long as we keep the powdered reagents separate from the liquid reagents during the irradiation and then mix them for analysis, everything is fine. We don't see any effects in migration time drift or difference in resolution between our peaks. And this just means that we have to have a sample handling system that can keep the liquid and the solid separate until we're ready to use it. And then the next thing we did, especially for the dye, we looked at the long-term storage at elevated temperatures. So So the dye is fairly sensitive to increases in temperature because the main degradation pathway is hydrolysis. So the hotter it gets, the faster that's going to happen. And the manufacturer recommends that you store it at -5C for 1 year. And that's too short for us. So we needed to make sure that it was going to be okay at higher temperatures for longer. So we stored it for 1 month, 6 months, 1 year, and 2 years at a variety of different temperatures. So we chose 4C, 25C, and 60C. You can see that at the elevated temperatures and longer storage times, you do start to get this increase in background. So that's the degradation of the dye, increasing the background. However, even at the worst case, with 60C for 2 years, we're still able to detect a majority of our amino acids, including glycine and histidine, which were two important ones for those biosignatures that we talked about. So CE is a great technique. It can provide chiral resolution and very low limits of detection. The chemicals needed to perform this assay are stable up to 300 kilorad, which makes them viable to fly just about anywhere. We will need a thermal control in the vault to keep the dye at a lower temperature so it doesn't start to degrade. But these are the first steps in making this technique more viable for future trip to Enceladus. So I want to thank the funding, and then also my group is here in force. So if you're interested in this stuff, please go and look at their posters and go watch their talks. Thanks. Yeah.
The mic works now. The mic works.
Oh, it works? Okay.
I think so.
Yeah. Richard Mathis, Chemistry and the Space Sciences Lab at UC Berkeley. The fly-through profile is very interesting, but of course one of the big challenges that we worry about, and I suspect you worry about, is how in the world do you capture the molecules in the plume, and what velocity do you transit through the plume? Okay.
Mm-hmm.
So what did you propose to do, and what are the issues and solutions to that issue?
So I can't actually talk about that because that's proposal specific, but we did propose something, and if if amino acids are there, we will detect them.
Oh, okay, that's all I get to find out.
Just a quick question: how much liquid sample would you need to do your analysis?
So the injection plug for capillary electrophoresis is about 4 nanoliters, so we only need a couple of microliters of sample in order to...
A couple of microliters?
Mm-hmm.
Okay, thanks.
Yeah.
Hi, I wanted to know if you test this dye against other nitrogenated compounds like amines,
Mm-hmm.
and if it's stable, like for example with alcohols and some other nucleophiles.
To be able to react with
Yeah.
other nucleophiles. So this is an amine-specific dye. I think it's... I don't think there's any cross-reactivity with like other nucleophiles, I'm pretty sure.
But
I mean,
for example, amines in a mixture with amino acids.
yeah.
Have you tested that?
Yeah, and so that's the benefit of separation.
Oh, cool.
Is that if there's a whole bunch of amines in there, we'll label them,
Yeah.
but then they'll get
Selected,
separated
yeah.
out from our region of interest.
Gotcha.
Yeah. Cool. Yes, let's thank Jessica one more time.